So in topic 21, we're going to be talking specifically about the substance of genes. In this Griffith experiment, he had two different bacterias, two of the same bacterias, different strains. Both strains grew really, really well, and only one of the strains caused pneumonia. That's the blue one. The red one did not. So with these disease-causing bacteria, the blue one, it was very smooth, so like a penny, while the harmless one had very rough edges, so kind of like a saw. So Griffith, he injected mice with the disease-causing bacteria. The mice all developed pneumonia and they all died. When he injected them with the harmless bacteria, they all stayed alive. So what kind of toxin is the blue bacteria, the S strain, producing that made them sick? So he ran a couple of experiments. First, he took the S strain, he heated them to kill them. Basically, it's the same idea as a fever when we talked about it in the immune system. It's going to denature or split the DNA apart. So when he heated the S strain, he killed it, the bacteria, injected it into the mouse survived. It shows it dead here, but that's not true. It's live. So it did not get pneumonia. We somehow killed the toxin. It was good. The mouse was alive. In the next experiment, he mixed the heat-killed one, so the one that did cause diseases that we heated to stop it, and he mixed it with the harmless one and injected it to the mouse. The mouse died, and it developed pneumonia before it died. So the lungs of the mice were filled with the disease-causing bacteria. How? How did this happen? So the reason it happened was basically something called transformation. So bacteria can talk to each other, and they can transfer the DNA to what specifically causes the toxin, which causes pneumonia and kills them. They were able to transfer that information and talk to the other bacteria and be like, hey, this is how you do it, kill the mouse. So it's called transformation, when one type of bacteria transforms into another. And this was not inherited by each bacteria in that specific generation, but it was actually passed on to all the offspring afterwards. So this transforming factor, he knew, had to be a gene. So a group of scientists at the Rockefeller Institute of New York, led by Oswald Avery, wanted to determine which molecule specifically in the heat-kill bacteria was most important for transformation. So here we're going to be talking about specifically the components that make up DNA. So DNA is a nucleic acid. Anytime in any test you see the word nucleic acid, it always relates to DNA. So DNA is made up of nucleotides that are joined together by long strands of chains by covalent bonds. So nucleic acids are long, slightly acidic, and identify in the nucleus. Nucleic acids are made up of nucleotides linked together to form long chains. So nucleotides look like this. You have 4 of them here. You have adenine, guanine, cytosine, and thymine. So the bases, the nitrogenous bases, are or nucleotides, are the AGCT, adenine, guanine, cytosine, and thymine. Now, in DNA, the word DNA stands for deoxyribose nucleic acid. The deoxyribose is this pentagon shape. It's a sugar. And what holds them together is these phosphate groups. That's what allows them to connect together. And it helps make something called the sugar backbone, which is what this is. And that's nucleic acids. So there's 3 components, like I said, 5-carbon sugar, phosphate group, and a nitrogenous base. And they're held together by covalent bonds between the sugar and phosphate groups. So these bases stick out sideways and form the nucleotide chain, and they're joined together in any order. So they can have any sequence. So they can be 12 A's, one after the other, and then 12 T's, or 12 G's, or 12 C's. It doesn't matter. It can have any order. Those orders are going to be specific to a trait, though, afterwards. So Erwin Chargaff discovered the percentages of adenine and thymine are almost equal to each other in any sample. So if you have 25% adenine, then you should have 25% thymine. And the same thing happens with guanine and cytosine. So if you have 25% guanine, you have 25% cytosine. So you can always figure out how much of each one there is, as long as you have one of the nitrogenous bases. So this is this observation shown there became known as Chargaff's rules. And the double helix model. So the double helix model explains his rules of base pairing. Base pairing is talking about, once again, the nitrogenous bases and how they match up. So, and it shows as a double helix, kind of like a spiral staircase, and how this model talks about how the two strands are held together. So it looks like a twisted ladder, like you grab a ladder and you twist it up. And in the double helix model of DNA, the two strands twist around each other. Now, this accounted for Franklin's X-ray pattern and explains the rules of base pairing and how the two strands of DNA are held together. So Franklin is Rosalind Franklin, and she actually she had an X-ray of it, which I'm going to show you guys right now. Nope. So this is Rosalind Franklin. This is a picture of her. This is her X-ray that was able to show the double helix model. It's like if you're looking at DNA from the top. And this is a picture of the DNA model. So it was able to explain how the two strands are held together. So each in the double helix model, the two strands run anti-parallel. So they don't move in the same direction, they move in opposite directions. So this allows the nitrogenous bases on both strands to come in contact right in the middle of the molecule, and they hold together with a hydrogen bond. So it's weak enough to pull apart whenever we're replicating, like we do in mitosis, but it's also strong enough to stay together. So Watson and Crick, after taking the information from Rosalind Franklin's X-rays, discovered that hydrogen bonds formed between them and provided just enough force to hold the strands together. They're weak enough, like I said, that they could be split apart, but they're strong enough to hold together for what is needed. Notice that guanine and cytosine have 3 hydrogen bonds, while adenine and thymine have 2. So with their model, it fit perfectly, and it allowed to and it showed the double helix model. So what role does DNA polymerase? DNA polymerase, the best way to explain it is to think of your English teacher. DNA polymerase is a proofreader. So it's an enzyme that's going to join the nucleotides to produce a new strand. So base pairing in the double helix explained how DNA can be copied or replicated. We talked about this in mitosis. Now we're going to specifically talk about how it replicates. So each strand of the double helix molecule has the information needed, and each strand can be used to make up the other strand, and the strands are said to be complementary. Once again, this is not complementary like, hey, you're gorgeous, or hey, you're beautiful, or you're awesome. This is complementary as it matches up. So every adenine is going to match up with thymine, and every guanine is going to match up with cytosine. So before cell divides, it has to duplicate its DNA. We know this is called replication. We call it DNA replication specifically in mitosis and meiosis. So this ensures that each cell has the exact same complete set of DNA molecules. So during replication, the DNA molecule separates into 2 strands. So it's going to split up like this and produce 2 new complementary strands following the rules of base pairing. So each strand of the double helix molecule serves as a template or a model for the new DNA strand. So in DNA replication, you're going to have this enzyme called DNA helicase that's going to split DNA just like this. And then you're going to have the DNA polymerase here that's going to come around, and it's going to help build all the other nitrogenous bases, and it's going to attach them to the ends. Primase is going to be leading it so it knows exactly where to go and which specific strand to go on to. So during replication, it's going to follow the exact same complementary rules, where every adenine is going to have a thymine and every guanine is going to have a cytosine, and they're all going to match up. So just like I said, the 2 strands in the double helix are going to separate. DNA helicase is the one that's going to separate them. It's going to split them up just like it is right here. So as each new strand forms, new bases are added. They are going to complement the old ones. So this is a original strand, and this is a new strand that's going to be forming. Remember how I said before that they are running anti-parallel. So this one is going to replicate this way. This one's going to replicate the opposite way. So at the end, we should have two exact same DNAs, exactly as the original one. So one is going to, like I said, it's going to be the original strand with the new strand. And then the other one on the bottom is going to be the original strand with the new strand. You can differentiate them in the photo down here with blue and orange. And they're going to be exactly the same. So DNA replication is carried out by a bunch of enzymes. The one that unzips them, once again, is DNA helicase. I showed you guys that image from Amoeba Sisters. The other enzyme is going to be DNA polymerase that's going to join them together. So it's going to build and it's going to proofread to make sure it's as perfect copy of the original. Now, at the end of chromosomes, there's these things called telomeres. At the end of DNA molecules, those are really difficult to copy. So picture that you make a copy, and then you make a copy of the copy, and then you make a copy of the copy. It gets messed up after a while. So that's what happens to the telomeres. You get some of those DNA molecules get lost over time, and since DNA is replicated so often, they get messed up. So an enzyme called telomerase, it compensates for this problem, and it adds short repeated. So it'll add like a bunch of A's or a bunch of T's or a bunch of D's or a bunch of C's to lengthen the chromosome slightly. And so it doesn't really matter that those telomeres are lost because they don't hold any important information. And how does this differ in prokaryotic cells? Basically, in prokaryotic cells, it's a circle. So the DNA in a prokaryote is a circle, and instead of it replicating like ours in two different directions, it just starts at one point and finishes it off. So actually it goes in two different directions until it finishes the circle. So in eukaryotic cells, replication begins at dozens or even hundreds of places. So that's like if we were all in the hallway and we were all working at different points in order to finish faster. So instead of starting in one spot and just continuing all the way down, because DNA is super long, it starts off in multiple spots. All of them work a bit in each one until it's completely copied. So the cells of most prokaryotes, like I said, are a circle of DNA molecule in the cytoplasm, and nearly all the cell's genetic information. Eukaryotic cells have 1,000 times more DNA, so it takes us a lot longer to replicate. So here, this is a prokaryotic cell. The blue is the original cell, the red is the new. This is the part where they're starting, and then they're going to split until they replicate everything. It's super easy because it's a prokaryotic cell and it's just a circle. In eukaryotic cells, like I said, it's going to go into different spots. So these are going in opposite directions, and so are these, until they basically meet at the middle, and then they go to another spot to then replicate that. So hopefully this gets copied very, very quickly. So 2 copies of DNA produced by the replication each chromosome remain closely associated until the cell enters prophase of mitosis, and at that point then they condense, you're able to see them, and then they start splitting afterwards with a complete set of genes coded with the exact same copy of DNA. And that's it.